Hello everyone, my name is Aditya Prabhakar and I am an undergraduate student at Georgia Tech studying neuroscience. Today I'm going to be starting my video course for molecular neuroscience, and we're going to start off by talking a little bit about cellular structures within a neuroscience realm. So let's get right into it and talk a little bit about cytoplasm. Now, bear with me, some of the stuff you may already know, but it's great to have a functional background before we get into the harder portions of this course. So, the cytoplasm is separated into three main portions: the cytosol, vacuoles, and ribosomes. For the purpose of this video, I'm going to skip over the cytosol as it's mainly considered to be either a placeholder or a background where you can find your vesicles, your vacuoles, and your ribosomes. So, important need to knows about vacuoles. There are many different types of vacuoles, and just to begin with, a vacuole is a membrane-bound organelle that is involved in storage. So I'm going to go down a list of some that I think will be important to you in your future journeys in neuroscience. So let's start off with the ER, the endoplasmic reticulum. This is where your macromolecules are synthesized. Then you have your Golgi complex, which is where proteins are processed before transport. You have your vesicles, which are your fluid-filled sacs that transport matter in and out of a cell. Within a vesicle, you have endosomes, which are those that transport molecules within a cell via endocytosis. And lastly, for the sake of the portion of vacuoles, I'm going to talk about lysosomes. And lysosomes is kind of similar to what it sounds like, a Lysol. So Lysol wipes are used to clean things, right? And they destroy bacteria. Similarly, lysosomes destroy the products within the endosome. Awesome. Let's move on into ribosomes. So the easiest way for me to explain the importance of a ribosome is kind of like telling a story, and that's how it was easy for me to remember it when I took a course like molecular neuroscience or even cellular and molecular biology. So the easiest way, like I said, is a story. So let's start off with inside our nucleus. Inside our nucleus, we have our DNA, which is now being transcribed into mRNA. From here, the mRNA is processed and finds a free ribosome to bind to. Protein translation occurs, and there is local protein synthesis in terms of a neuroenvironment. The synthesis usually occurs on dendrites that are near synapses. From here, protein translocation sends the protein to the ER, forming the rough ER. Then we move on to the Golgi apparatus, and then from the Golgi apparatus, we go to exocytosis, and the protein or whatever matter that you're making exits the cell. Awesome. Let's move on into the cytoskeleton. So now that we've closed off the cytoplasm, we're moving on to the cytoskeleton. Cool. So for the purpose of this video, we're going to talk about three different portions. You have your microtubule, your neurofilament, and your microfilament. I'm going to put an image or a visualization on the screen right now to kind of show you the differences between all these three. And for this time, I'm going to let you know that the most important thing you should be remembering is the size, the actual function, and if it is polar or non-polar. Awesome. Axonal transport. Axonal transport is how proteins are moved along microtubules and actin filaments. So it's really important to know that it's a different format in axons and dendrites. And before I get into actual axonal transport, I'm going to fast forward a little bit into the anatomy of a neuron. So a neuron is created not symmetrically. So it's asymmetrical in the way that it has your neuron head here, your axon, and your dendrites. And the way this works is your dendritic spines are the ones that send out information to other neurons and contain a lot of the smooth ER and polyribosomes to have the synthesized proteins ready to go. Your axon itself is rich with synaptic vesicles because this is where transport occurs within a neuron. The distribution of all these different vacuoles, such as the ER, Golgi complexes, and lysosomes, decrease the farther you get away from your head, otherwise known as your cell body. Awesome. So let's go back into axonal transport now. We were talking about the difference between axonal transport within axons and dendrites. So within the axon, you're going to be moving from a plus end, which is towards the synapse, and the minus end, which is towards the cytosol. In dendrites, these plus and minus ends are very mixed up, and there's no real organization or configuration for knowing what is what. Awesome. So we have fast axonal transport and slow axonal transport. An important acronym you should be knowing for fast axonal transport is MCARD: myosin and kinesin, anterograde, retrograde, dynein. So anterograde and retrograde, what do those two words mean? Anterograde means transporting from the soma down to the axon, and retrograde means transporting from the axon to the soma. Again, the acronym is MCARD: myosin and kinesin, anterograde, retrograde, dynein. And we have slow axonal transport, which is used for protein transport, and it's only anterograde. Awesome. So the last portion of this video is glial cells. And it's important to not misdescribe a glial cell as an actual neuron, because neurons are excited electrically while glial cells are not. So there are different types of glial cells, just like there are different types of vacuoles and other types of organelles. The glial cells that we're going to talk about, we're going to separate it into two categories: the CNS, which is the central nervous system, and the PNS, the peripheral nervous system. Within your CNS, you have oligodendrocytes, astrocytes, microglia, and ependymal cells. I'm going to list all the differences here, and I will let you guys know another acronym towards the end of this section to describe how you can remember the CNS versus the PNS. Awesome. And within your PNS, you have your Schwann cells and your satellite cells. Let's roll it back one more time. Within your CNS, you have your oligodendrocytes, which are used to produce myelin, your astrocytes, which regulate your neurotransmitter concentrations within your synaptic cleft, your microglia, which are involved in immune support, and your ependymal cells, which are, it's kind of an everything cell that deals with CSF production, which is spinal fluid, waste clearance, and brain metabolism. Within your PNS, you have your Schwann cells, which produce your myelinated sheath, and your satellite cells, which again are an everything cell that kind of just support your system with providing nutrients to surrounding neurons and extra cushioning for protection and stuff like that has to deal with pain. Now, the acronym is COMPSS, COMPSS. The CNS deals with the oligodendrocytes and the microglia, and the PNS deals with the Schwann cells and satellite cells. Thank you guys for bearing with me through this video, and I hope you enjoyed it. Again, if you'd like to see more videos, please like, comment, and subscribe, and I'll see you guys in the next video when we talk more about membrane transportation.